Hi everyone, welcome to today's lecture. Today we are going to talk about microbial ecology. So today's objectives are eight. First, we will name four major contributors to the carbon cycle. We will define the methanogenic. We will define methanogenic and explain the role of methanogens within the carbon cycle. We name and summarize the four reactions involving the nitrogen cycle, including the starting and ending product. We will describe the relationship between rhizobia and legume. We will define microbial ecosystem and discuss one example. We will explain eutrophication, eutrophication, and specifically how microbes are responsible for its impact on aquatic life. We will define methanogenic sampling and define examples how it have changed our view of microbiology, and define One Health and how it impacts diseases. So basically, this is the outline. We are going to talk about the biogeochemical cycles, which is basically carbon cycle and nitrogen cycle. And then we will talk about microbial systems and the One Health. So how do microbes obtain nutrients? Depending on the microbial type and where they grow, they obtain nutrients in the form of carbon, oxygen, or nitrogen. And now let's take a step back and consider nutrients at the ecosystem level. So this is basically through the biogeochemical cycling of nutrients, and then we will talk about the important roles of these microbes in recycling this. So element cycle between living and non-living organisms. I'm sorry, between living and non-living environment. So basically, living environment gives off some elements to the non-living, and the non-living give back to the living. And cycling and recycling maintains the necessary balance of nutrients so that both sides can get what they need. So cycles are complete systems that rely on interplay of different type of organisms. All organisms participate in recycling, but only certain type of microorganisms have the metabolic pathway to convert inorganic compounds from one nutritional form to the other. So first, let's talk about one of the major cycles, which is the carbon cycle. So carbon exists as carbon dioxide in its nature form, and it's been seen in mineral state and as an inorganic reservoir in bodies of organisms. So basically, when you die, you decompose, you give off carbon. So microbe plays a role in carbon dioxide cycle. Basically, what they do is that they recycle through the ecosystems through respiration, fermentation, carbon fixation or photosynthesis, and also the production of methane. So first, let's talk about methane in the carbon cycle. I mean, what I talked of basically is what is here. So let's just go to methane cycle. So methane gas play secondary part in the carbon cycle. More potent greenhouse gas. It's a more potent greenhouse gas than the carbon dioxide. I think there's been several laws being made to basically reduce the amount of methane produced. But the reason why laws are being made to regulate methane production is that they can trap nearly 20 times more heat in the atmosphere. For that reason, releasing more methane in the system is very bad. But the downside is that methane is actually released from GI gut of ruminant animals and swarm gases. So unless we cut down the amount of ruminant we rear, and basically the meat we consume, it's very hard to bring this down. And also, it's being also produced from cars, engines of cars. Methane gases are sometimes produced from there. No, no, I'm wrong. No. So, still on methane. So, some microbes called methanogens basically have the ability to regulate produce methane. So these are archaeas or archaeas, and they live in the anaerobic ecosystem, basically in ruminants, swamp, humans, and more. And they convert carbon dioxide and hydrogen gas into methane. And below here is some example of them, which is methanopyrus. And this is methanobacterium and methanococcus. They are found in a wide range of temperatures, pH, and salinity. So now we've discussed how methane is being formed. Let's move into the nitrogen cycle. So the nitrogen cycle is more intrinsic than other cycles. Um, it includes multiple reactions that have to occur for it to be fully complete. Higher plant utilizes nitrate and ammonium ion, and then microbes use all these forms of nitrogen to give us what we need. So there are basically four types of reaction that have to happen within the nitrogen cycle. We have the nitrogen fixation, ammonification, nitrification, and denitrification. And basically, as you've seen from here, this is when nitrogen fixation come in, where nitrogen gas is trapped into the soil, and from there, second part happen, nitrification, converting of nitrate to nitrite, and then plant just absorbs it, and then the next stage happens where nitrogen is made available to everybody. Then ammonification also happens, and then the whole thing goes on where lastly denitrification also happens. So now let's talk about nitrogen fixation. So as I explained earlier on, atmospheric nitrogen is converted to ammonia and is performed by bacteria and archaea. And this there are enzymes which are involved in this complex process, basically converting this. These enzymes are called nitrogenase, which helps in the converting of nitrogen to ammonia. And they are mostly present in soil and whether aerobic or anaerobic environment. And they are some of them are present in some free living organisms, and some also have a symbiotic relationship with plants. In such example is Azotobacter, which is present in aerobic free living, and we have the Clostridium species, which is present also in anaerobic free living. So there is a these these enzymes, or basically this substrate or enzymes that help in the converting of ammonia to these bacteria that helps in the conversion of ammonia to atmospheric ammonia to ammonia in the soil. They mostly have a symbiotic relationship with plants. One of these examples is the Rhizobia species. This Rhizobia species bacteria have a close relationship with legumes, preferably peas, beans, and clover. Basically, they helps in the conversion of ammonia to for these plants, and these plants also give back to them by providing nutrients. So the Rhizobia species, or the Rhizobia, is a gram-negative bacteria which is motile and also bacillus in nature. They help colonize legume roots and produce root noodles. Supplies reduce nitrogen or ammonia to these plants, and then the plants also give back to them. And one example is what we've actually shown here in this picture. So now we've talked about how ammonia is being converted. Now we have to talk about ammonification. So ammonification happens through the decomposition of organic matter to ammonia and then ammonium ion. So then we have the bacteria such as Bacillus and other species contributing to this, and some fungi also contribute to this as well. And as you can see in a nice illustration here, this cow is giving off organic nitrogen, and then the bacteria work on it, producing ammonia. So now we've talked about ammonia, nitrogen fixation from the sky. We've talked about ammonification, which is basically happening through the defecation of the cow. Now we have to talk about nitrification and then lastly denitrification. So nitrification is basically conversion of the ammonia which have been given off to nitrite. So nitrite basically is the most oxidized form of nitrogen used by plants. And this is carried out by mainly bacteria. So it include two steps. We have bacteria such as Nitrosomonas, which oxidizes ammonia to nitrite. Secondly, we also have the bacteria such as Nitrobacter, which also oxidize the nitrite to nitrate. So some fungi and archaea contributes to this. So as we've shown here, conversion of the ammonia oxidizer, converting the ammonia to nitrite, and then converting it to nitrite, and then from nitrite moves to nitrate, also by a different group of bacteria. So one thing you have to note is that the obligate aerobics. I'm sorry. So one thing you have to know is that this procedure is obligate aerobes, so they cannot happen in waterlogged soils or anaerobic aquatic environment. Nitrate is used as a terminal electrode acceptor in the anaerobic respiration. So this whole procedure is actually super important. And then lastly, let's move to the denitrification. So denitrification basically means the conversion of nitrate to nitrite through immediate steps to atmospheric nitrogen gas. So this is carried out by hundreds of different species of bacteria and then contribute to nitrogen loss in the soil, which in some cases is bad for plants or crops. So basically, we have nitrate here, and then it's converted to nitrite, and then nitrite reductase, which is the bacteria, helps reduces it back to nitrite, and then the nitrite oxide helps reduces the nitrate back to nitrous, and then the nitrous oxide helps release it back to nitrogen gas and is sent back to the atmosphere. This whole procedure happens in an anaerobic environment. So you can have some time to practice the microbial ecology case. And then you can also practice this too. So now that we've finished talking about the biogeochemical cycle, we have to talk now about the microbial ecosystem and then the One Health. So microbial ecology basically is the study of microbes in natural habitat, and we study this organism in isolation, and but they are never in isolation. I think we've practiced few of these. And basically, when we talk of an ecosystem, this is basically a collection of organisms together with the surrounding physical and chemical factors. It can be biotic and abiotic factors. So now let's talk more about the ecosystem and where we can find these microbes. So we can find some in aquatic environment, and in the aquatic environment we have different kinds of microbes. Some examples include the plankton. Basically, these are floating community that drift with waves and current. And as you can see them here, they are present in aquatic water bodies or water bodies, and just this basically here is just a group of microbes, and they are all floating around. And we have phytoplankton, which are photosynthesis algae, and cyanobacteria, which helps basically photosynthesis algae helps produce photosynthesis in the presence of light. And we have zooplankton, which are microscopic consumers such as protozoa and invertebrates. These are actually great abundance of bacteria in the aquatic environment, and there are also presence of some viruses too. You can try and a study done by J. Crick Venter basically did an expedition where they did a sampling of oceans from 2004 to 2007, and they discovered about 6 million new genes and 10,000 of new proteins. So you can appreciate how wide these microbes are present all over in the environment, including the aquatic environment. Please try and watch these 4 videos, and get a good look of it. So we have eutrophication, basically addition of excess nutrient to aquatic ecosystem. So the first step is just point one is a heavy surface growth of cyanobacteria and algae, which shut off oxygen supply to the lake. As you can see, they are just growing all over the place, and then there's not enough oxygen going down into the lake. Then aerobic heterotroph depletes oxygen further by decomposing the organic matter, causing massive die-offs of straight aerobics, example fish and invertebrate. And then only the anaerobic or facultative organism can then survive. And this whole situation actually becomes worse by sometimes the addition of industrial waste, detergent from household waste, and runoff of manure or fertilizers into this, which mostly happens, when people live closer to lakes and farmlands. And this can just make the whole situation super worse. So let's talk about the soil environment. So about 70% of the whole earth, bacteria and archaea lived underground. And some examples include mycorrhiza, which is which is a type of bacteria that we have. And these bacteria actually have a symbiotic partnership between vascular plant root and fungi. But basically, in the soil environment, there is a dynamic ecosystem with many microbes. About 1 gram of soil have about 10 billion microbes, so you can actually appreciate how complex it is. And because of the porous nature of the soil, there is also presence of numerous microhabitats. We have aerobic and facultative organisms occupying looser and drier soils, and anaerobic um, organisms that are adapted to waterlogged and poorly aerated soils. Deep surface sampling, approximately greater than 2 miles below the surface, shows that bacteria are found 30 meters or 90 feet below sea floor in clay. Discovering new metabolic capabilities in deep surface microbes. So you realize that even as deep as over 2 miles down the surface, there's still bacteria or microbes still present down there. So we've talked of the soil microenvironment and how bacteria are still present in there. Now let's talk of the human microenvironment. So the human body is also an ecosystem. Basically, we harbor microbes in our gut. The microbiome, which many different the human microbiome consists of many different microbial types, which are found through part of the body, and there's a lot of factors that contribute to their composition. For example, in the esophagus, the microbes present there needs a pH of 4.0, and that of the colon also need a specific pH. The stomach will need a specific pH. So depending on pH and the other environmental factors, that will determine how much microbes you'll be having in your gut. So how do we know which microbes are present? That brings us to metagenomics. Basically, this changes how we view the environment. So in order to know how many microbes are present, we can follow this diagram where DNA is isolated and fragmented and filled with adapters. So basically, you sample the environment, get some samples, you isolate DNA from them, you break the DNA down, fragment them, you attach adapters to it, then you send it for sequencing. You PCR it, and then So wait, let me take it again. So now we take the samples out and then we isolate the DNA, we fragment it, we add our adapters. The adapters are added to the PCR machine. And then the resulted amplification is being sent for sequencing. And then after sequencing, we can read them and then we can align them using softwares. And then we determine how much variation that we have or how much microbes that we have been present. And through this method, we can identify new microbes in new places, and we can also understand the abundance and diversity. And then we can also identify novel traits or genes in these microbes. Basically, genomics has changed our view of microbiology, which helps us understand fully the diversity and abundance of these organisms. So, what is the concept of One Health? So microbes, as we've learned today, circulate among human hosts, animal hosts, and environmental reservoirs. So these then changes in this environment can lead to transition of pathogens to animals and humans that previously were exposed to them. So you can see that from this diagram, for example, this pathogen can infect or can be present in both chicken, humans, and plants. So basically, pathogen evolution, these changes in the changes in either of these environment can assist in the pathogen evolution. Basically, the evolution of this pathogen for it to better adapt to either the chicken, plant, or humans. Basically, adaptation of different hosts. And if this happens, this will cause the increase in disease incidence. Basically, this pathogen will have the ability to infect chicken, humans, and then plants. And then this will also result in changes in disease location. One example is change bird flu and COVID-19, where we had the SARS-CoV-2 coming back again, and sorry, we have bird flu changing to different states, and also we have SARS-CoV SARS state. We have SARS then changing to COVID-19. So an example of this again is the hantavirus acute respiratory distress disease. This is common in southwestern US and population growth of deer mouse following, and an example is also population growth of deer mouse following El Nino associated heavy rainfall in 1993. This results in decrease of deer mouse predators, increased growth of peanuts, and then a favorite food source. So these guys ended up dying and then resulting in a lot of food source. So we have increase in malaria in East Malaysia. Um, Deforestation, it brought when trees were cut, or the whole deforestation brought organisms closer together, and then resulting in the plasmodium or the female Anopheles mosquitoes having access to these people and transmitting them. Then yeah, basically giving access to these mosquitoes, biting both monkeys and also humans, transmitting disease to these two organisms. So today, this will be the end of lecture for today. Today was super quick. Please see you tomorrow. See you on Friday, and then come super prepared. Bye.